Hi, hello, good evening everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. V-Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, recorded sessions, study materials, concept videos. Moreover, you're going to get this lecture notes in the form of PDF. Get your acquaintance with all the channels available and subscribe to the channel Vistas Learning Class 11 and 12. If you find this session useful and interesting, make sure to click on the like as well as share this session with your friends. And also click on the bell icon so that you are going to get all the upcoming video notifications. Now, let's begin our today's session. We are in a series of the sessions where we are discussing about the chapter Molecular Basis of Inheritance that comes under the unit Genetics and Evolution. Let's start now. So, in the previous session, we discussed about the semi-conservative mode of DNA replication, right? So, today, here is an experimental proof to prove that the DNA replicates in a semi-conservative way. What do you mean by this semi-conservative? So, we already know that DNA in its double-stranded form, it is first going to separate to form single strands. Now, the single strand is going to act as a template for forming daughter strands. So, using the single strand of the parent DNA, this is a parent DNA. Now, this parent DNA separates to form single strand. This strand is acting as a template strand. This strand is going to act as a template strand which is useful to give the information, and thereby a new strand is produced here, and this one is a newly synthesized strand. New strand. So if you consider daughter DNA, we have one strand coming from its parent DNA and other strand which is newly synthesized. So because of this reason, where one of the strand is conserved, that is what we call it as a semi-conservative mode of reproduction. Semi-conservative mode of DNA replication. Now, this is now proven that the DNA is going to replicate in a semi-conservative mode. It is proved by the experiment done by Michelson and Stahl. It was now shown that in E. coli, Escherichia coli, and subsequently, starting with the prokaryotic simple organization to the eukaryotic, that is higher organisms such as plants as well as human cells. And just now we said that the semi-conservative mode of DNA replication was proven experimentally, and that experiment was done by Matthews, Michelson and Franklin Stahl, which is popular as Michelson-Stahl experiment. They performed an experiment in 1958. Let's see what experiment that they have done to prove that the DNA is going to replicate in a semi-conservative mode. So they have allowed Escherichia coli to grow in a medium, and this medium is provided with ammonium chloride. Here, this ammonium chloride is of 15 nitrogen containing, and this nitrogen 15, it is a heavy isotype. Here, this is very important that this is heavy isotope but not a radioactive isotope. That is what radioactivity is not used. Instead, centrifugation technique is used to separate and to detect the distribution of the DNA that is formed in the next generation. So here, what happened? They allowed actually Escherichia coli to grow on a medium, and this medium is containing N15. That is, N15 is incorporated into the ammonium chloride, which is acting as a source for the nitrogen. And just now we said that this N15, it is a heavy isotope. That means based on density, we can separate it. And this is the nitrogen, which is the only source that is available. Therefore, it will get incorporated into the DNA that is present inside the each and every Escherichia coli cells. So here, they have taken a petri dish to which the medium is provided, and this medium is containing that nitrogen heavy isotope containing ammonium chloride. So, whatever the nitrogen compound which is going to be formed in the cells of the Escherichia coli, that is going to incorporate with heavy isotope of nitrogen. So, here the cell starts growing. Cell starts growing here. If you take each cell, inside DNA replication takes place. And for the newly formed DNA, the nitrogen source is heavy isotope nitrogen. So here is the point. The result was that N15 was incorporated into the newly synthesized DNA. Newly synthesized DNA as well as other nitrogen containing compounds like proteins. So here the point is very simple. The Michelson and Charles experiment, where they have conducted by taking the Escherichia coli, and they have grown this Escherichia coli in the medium where they have provided the heavy isotope nitrogen containing ammonium chloride. So, the growing Escherichia coli starts on incorporating this heavy isotope of nitrogen, as this is the only nitrogen source that is available in the medium. So whatever the newly synthesized DNA, it is going to take it is going to incorporate with N15. Now, here is a point. N15 is a heavy isotope, and N14, it's a normal isotope. But these are not radioactive isotopes. This is important. These are not radioactive. Okay. So, this heavy DNA molecule, why we are calling it as a heavy DNA molecule? Because it got incorporated with the N15. Whatever the cells that are growing, inside the synthesis of the DNA takes place, where the DNA utilizes N15 as a source of its nitrogen. Therefore, the formed DNA, we call it as a heavy DNA molecule. And this heavy DNA molecule could be distinguished, made separate from the normal DNA. This is what N14 is, a normal DNA. And that separation, that distinguishing is made possible by using the technique, and that technique is called as centrifugation. DNA by centrifugation in a cesium chloride density gradient. And this technique here we are using because the isotope is heavy isotopes. What we have used are the heavy isotopes, but not the radioactive isotopes. If we have used those radioactive isotopes, then we have to go for the radioactivity techniques. But here we, based upon the density we are separating, that is what we have taken here, heavy isotope. This point is very, very important. And please note that N15 is not a radioactive isotope and it can be separated by N14 only based on density. That is what we are using, density gradient cesium chloride centrifugation technique to separate N15 DNA molecule from the N14 DNA molecule. Now, so here, then they transfer, see, first they made to grow on the medium which contains N15 ammonium chloride. So here we have the cells which produces DNA, and the DNA gets incorporated with the N15. Now they have transferred the cells into another medium. Now the new medium contains N14, which is a normal nitrogen, NH4Cl. Now the cells has been inoculated from this medium to the new medium. The new medium is incorporated with the normal nitrogen NH4Cl medium, that is ammonium chloride medium. So here then they transferred the cells from the medium, from this medium into a medium which contains normal N14 containing ammonium chloride. So here, and after the cells started growing in the new medium, then they have taken the samples. They took the samples at a definite time intervals, various definite time intervals. Meanwhile, the cells multiply. That means the cells doubles its number. Doubling of the cells takes place. And extracted the DNA that remains as double-stranded DNA. So now, see, previously, the Escherichia coli had their growth in the medium in which N15 is a nitrogen source. Then they have transferred to the new medium where the N14 is a nitrogen source. Now here also cell starts dividing, cell starts growing, cell starts showing DNA replication. Now here we have taken the samples at a regular intervals of time, and thereby after taking the samples, DNA extraction was done. And that too, it remains as the DNA that remains as a double-stranded helix. Now what happens? The various samples which has been collected from the both the mediums were separated. Just now we said that the separation happens by using a technique, and that technique is called centrifugation technique. Cesium chloride density gradient. That is what we are, depending upon the density, that is what we used heavy isotope instead of radioactive isotope. This point is really important. Independently on cesium density gradients to measure the densities of DNA. Densities of DNA. Now, so here, this is the first sample where DNA is synthesized by using N15 as its nitrogen source. So both the strands of the Escherichia coli, that means both the strands of the Escherichia coli DNA will show nitrogen 15 in it. So if it is separated by using the cesium chloride density gradient centrifugation technique, we do find the formation of the band towards the bottom of the centrifuge tube. Why? Because N15 is heavy isotope. And this separation is based on the density gradient. We got the band towards the bottom of the centrifuge tube. Both the strands are made up of N15, 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 N15. That is what we consider this as a heavy DNA. Now, after 20 minutes, this is the generation time of Escherichia coli, where doubling of the cells takes place. That means duplication takes place. If one cell is present, it is going to divide into 2. After 20 minutes, 2 cells are going to divide into 4. And after 20 minutes, where the division takes place, one cell divides to 2. And also, the DNA replicates to produce 2 daughter DNA. Now here, as we already said, they transferred into a new medium where in the new medium, the nitrogen source is a normal nitrogen, that is N14, right? So here, whatever the newly available nitrogen is of N14 type. As this is semi-conservative way where the mother parental DNA divides to form single-stranded DNA. Now, the newly synthesized DNA has to form taking this parental strand as a template strand. Now, whatever the newly synthesized DNA will utilize the nitrogen source which is available. It got N14 as its nitrogen source. Therefore, if it is separated by again subjecting to the centrifugation technique, we'll get a band in between. It is not towards the bottom, nor it is not towards the top of the centrifuge tube, but instead we'll get a band which is formed in between. That means it is a combination of N15, N14, N15, N14, right? So, N14, N15, it's an hybrid band, hybrid DNA or intermediate DNA. Now, after another generation, this is all about first generation. Now, generation 2. Here also, this 2 strands now separates into 4 single strands, right? See, this will separate here and this will separate here. I will explain. Green color, it is a newly synthesized DNA in this generation, right? And it is going to act as a parent strand, template strand for the next generation, right? So first, this DNA separates and, right? Same thing happens with this strand. Right? The newly synthesizing DNA starts producing over this template strand, using this template strands. Now it is represented with the green color, right? Right? We got 4 newly formed DNA, daughter DNA. All these are daughter DNAs. Now, towards the end of the generation 2, we got 4 daughter strands, 4 daughter DNA, out of which this daughter DNA contains both the strands which are made up of N14, right? And this is the daughter DNA which got both the strands made up of N14. And these 2 daughter DNA is a hybrid in which one strand is made up of N14 and other strand is made up of N15. There are 2 hybrids and 2 light chains newly formed in the medium. That is the medium where the nitrogen source is N14. I'll write it clear. N14. Right? Same way, if these DNA, daughter DNAs are subjected to the centrifugation, then we'll get 2 bands. The same intermediate band where we got here, again it will repeat because 2 DNA are of hybrid types where one strand got N15 and other strand got N14, right? Now these 2 DNA strands where both two strands comes from N14, that is normal nitrogen. Therefore, these 2 DNA are going to form a new band here. This is already seen. It repeats. This is a new one where it is represented with light DNA. We got 2 light DNAs and 2 hybrid DNAs. So we got 2 bands in total, hybrid band whereas light band. So by this experiment, it is proven that the DNA replicates in a semi-conservative mode of replication. Similar experiment also done, and this is a theory, just to recollect it. Thus, the DNA that was extracted from the culture generation 1, first generation, after the transfer from N15 to N14 medium, that is after 20 minutes. So here, this is the DNA which is formed in the medium that is provided with N15. After 20 minutes, the cells are transferred to the another medium where the medium contains N14. Because of that, they form an hybrid of having one strand containing N15 and another strand containing N14. This is possible because it is replicated in a semi-conservative mode. Because of that, it is getting one strand from the parent DNA and one other strand is newly formed, newly synthesized. That is what, one strand is totally getting N14 because it is newly forming, and the remaining strand, that is the other strand which is coming from the parent DNA, which is considered as a template strand, that it is having the old N15 itself. If the newly synthesizing DNA, both the strands are newly synthesized, then we do have to find that both the strands should incorporate with N15, but that is not happening because it is semi-conservative, which conserves semi-part, that is half part from the parent generation. And Escherichia coli divides in 20 minutes. That is what generation happens. One cell divides to 2. Had a hybrid. This is what hybrid we call it as an, or intermediate density. That is not towards the bottom of the centrifuge tube or towards the top of the centrifuge tube. In between we got the band, and that is called as intermediate density. Now, DNA extracted from the culture after another generation, starting it is 40, and starting from the generation 1, it is after 20 minutes. That is after 40 minutes, that is second generation, initially from initial point, was composed of equal amounts of this hybrid DNA and a light DNA. Out of 4, 2 are hybrid, 2 are light DNA. So this is the proof, to prove that the DNA is replicating in a semi-conservative mode. Now, the same experiment was done by Tyler et al. And his colleagues in the same year, 1958. So here, very similar experiment involving the use of radioactive thymidine. There, they have used heavy isotope of nitrogen. Here, they have used radioactive thymidine to detect the distribution of newly synthesizing DNA in the chromosome. They have conducted this experiment on a plant, and that is faba beans. Visia faba. What they done? They have taken faba bean plant, and they provided to the root system of this faba bean plant with radioactive thymidine. So here, after some time, they have withdrawn this radioactive thymidine. Now, what happens? In the first generation, if the cells of the plants were taken and how the chromosomes are dividing, if you look, even the chromosomes are also going to divide by a semi-conservative mode. The experiments proved that the DNA which is present in the chromosome also replicates semi-conservatively. The DNA which is present in the chromosomes also do replicate by semi-conservative method. So this is how, if you consider this red color for the radioactive thymidine, this radioactive thymidine is going to segregate semi-conservatively. That is what, the one strand is completely of the parental strand, and the other strand is going to form newly. That is what, it is devoid of radioactivity. Now, so let's move to understand the enzymes and the machinery that is going to involve in the DNA replication. We know that the DNA replication takes place in prokaryotic cell as well as simple, and they don't have a well-developed nucleus, true nucleus. Whereas a well-developed nucleus can be seen in the case of eukaryotic cells. Therefore, the complexity in the replication of the DNA is more in the eukaryotic cell when compared to the prokaryotic cells. And now, in living cells such as E. coli, Escherichia coli, the process of replication requires a set of catalysts, enzymes. Not only enzymes, we need protein stabilizers, protein factors, and metal ions. Even the list of the enzymes is also goes like DNA polymerases, DNA helicases, DNA primerases, topoisomerases, and this all this combination, that is the enzymes, protein stabilizers, protein factors, and even metal ions all together are needed to perform DNA replication. Now, the main enzyme, important enzyme, is referred to as DNA dependent DNA polymerases. DNA dependent DNA polymerases. If you look at, this is a parent DNA. If you consider this as a parent DNA, we know that the parent DNA is going to unwind. Uncoiling takes place such that it is going to separate into single strands. While this is happening, this single strand is going to act as a template for the formation of newly synthesized strand on it. So, this newly synthesized strand, it requires enzyme, and that is called a DNA polymerases. As the DNA polymerizing is depending upon the parent DNA strand, that is what we call it as a DNA dependent. Here the DNA is a parent DNA. The DNA polymerase enzyme depends upon the parent DNA to take it as a template and to produce a new DNA, new daughter DNA. That is what the name is given as DNA dependent DNA polymerase enzyme. Since it uses DNA template to catalyze the polymerization reaction of deoxynucleotide. Deoxynucleotides. Right? These enzymes are very highly efficient enzymes as they have to catalyze polymerization at a very fast rate and with an 100% accuracy. Polymerization of a very long number of nucleotides in a very short time. If you look at the length of the DNA that is present in the Escherichia coli that has only 4.6 into 10 to the power of 6 base pairs. 4.6 into 10 to the power of 6 base pairs. This, when compared to the human diploid content, which is 6.6 into 10 to the power of 9 base pairs, it is very less. And to complete the process of replication within 18 minutes, that means average rate of polymerization has to be approximately 20,000 base pairs per second. The DNA polymerases has to attach 2000 base pairs per second. See, 2000 pairs of nucleotides. Pairs of nucleotides is nothing but one base pair. Such 2000 base pairs per second. This is the speed at which the DNA polymerizes. That means addition of the nucleotides takes place by using the parent DNA as a template. Now, here, not only do this, polymerizes has to be fast. Just now we explained the numericals such that it is fast enough. It has to add of 2000 base pairs per second in Escherichia coli, but they also have to catalyze the reaction with a high degree of accuracy. Not only it should work fast plus effective and have to be accurate. If the accuracy is missing, then it is going to show new changes, sudden changes, that what we call it as a mutations that shouldn't be happen. Any mistakes, any errors during replication would result in the mutation, very deteriorating condition towards the DNA structure. Furthermore, DNA replication requires, it is a very expensive procedure, it requires ATP. Energy is utilized. Energetically, replication is a very expensive process. It requires ATPs. Now, so here, deoxyribonucleoside triphosphates deoxyadenosine triphosphate, deoxythymidine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate. This will undergo phosphorylation reaction to produce energy. Just now we said that DNA replication is a very energetically expensive process because it requires more and more of energy, and that energy will come from the phosphorylation reaction where conversion of a triphosphates into deoxyadenosine monophosphate, deoxythymidine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate releases energy, and that released energy is going to utilize for the DNA replication, right? In addition to acting as a substrate, see here, all these deoxyribonucleoside triphosphates, besides acting as the substrates, they provide energy for polymerization reaction. Where it is getting from? From the terminal two phosphates. Here is a base, right? And this is the sugar which attaches to I'll take another color. Triphosphate, three phosphates, right? This terminal two phosphates, breaking of this thing, like triphosphate to monophosphate, it releases energy. This is the reaction where liberation of the energy takes place, and that energy is useful for the polymerization reaction. The two terminal phosphates, these are the two terminal phosphates in a deoxynucleoside triphosphates are high energy phosphates, same as in the case of ATP, right? Now, in addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. Just now we said that not only DNA-dependent DNA polymerases enzyme, always polymerization starts from 5' towards 3'. Always the polymerization reaction starts from the 5' to the 3' end. This is very, very important. As we already know that the double helical parental DNA splits, opens up, uncoils in order to form the single stranded DNA. Now, this single strand where it gives a shape of Y, this structure, we call it as a replication fork. And always the polymerization takes place in the direction of 5' to 3'. Look at the first strand of the parent DNA. It is moving from 5' to 3'. And therefore, here we can find 3'. Always the polymerization takes place from 5' to 3'. So, addition of the RNA primer here will leads to the continuation of the polymerization from 5' to 3', exactly towards the direction of the polymerization, that is 5' to 3', 5' to 3'. That is okay. Coming to the next strand, here it is running from 3' to 5'. And here we can see 5' over here. If it has to start the polymerization in this direction, it can't go parallelly to the template strand. Therefore, it has to start from an other end. From here, it starts synthesizing. That means addition of the nucleotides takes place. As it is running opposite, The polymerization reaction is not continuous, it is going to break, form of fragments, and that is what we call it as a discontinuous strand, whereas this strand is continuous strand. So here, along with the DNA dependent DNA polymerase enzyme, we have an other enzymes also which are needed. First and foremost, to open up the coiling, unwind, we need DNA helicase. And for synthesizing of RNA primer, we need DNA primase enzyme. Just now we said that unwinding of the helical structure of parent DNA takes place, which leads to supercoiling, and it will create the tension. To release this tension, again we need an enzyme, and that is topoisomerases. We said that one strand continues in a leading strand, continues as a leading strand, and the other strand will be interrupted, and we see the fragments over it, where we can consider this strand as a lagging strand or discontinuous strand. Why we are calling it this strand as a discontinuous strand? Because fragments are forming, and these fragments has to be connect, joint, link with each other, and that ligation happens in the presence of DNA ligase. So, not only DNA dependent DNA polymerases, there are many of the other enzymes are also required for the DNA replication. Just now we mentioned DNA primase, DNA ligase, DNA helicase, topoisomerases. Now, for long DNA molecule, complete unwinding to release into single strands is not possible because it requires a more and more of energy. Since the two strands of DNA cannot be separated in its entire length, it can't separate over its length, entire length. Due to very high energy requirement, the replication occurs within a small opening of DNA helix. Small opening of DNA helix. I'll show here. Consider this is a DNA. Unwinding completely to form entire the length to form a single strand of DNA, it is not possible because it is going to utilize a lot of energy. High energy is required. Therefore, it is not going to be separate entire of its length, but instead it will show small opening. Remaining will be in the coiling position itself. See, toward the length of the DNA, it is not going to be open. It is going to show a very small opening where the new strand starts forming like this. So here you can look like a Y-shaped structure, and that is what we call it as a replication fork. Replication fork. This is what. Here it will be coiled, and the other side also replication fork will form, and the other side also it will be coiled only. Whatever the part it is going to undergo, it is going to show the DNA replication, that is undergoing that small opening, and the remaining length of the DNA will be in a coiled position, that is helical position. Now, and that is refers to as a replication fork. Just now we said. Moving on. So here, the DNA-dependent DNA polymerases catalyzes polymerization only in one direction. This is very, very important. Just now I have explained 5 prime to 3 prime. That is what this is continuous. And the other side, as the polymerization doesn't happen, it starts to produce fragments, and that fragments are called as Okazaki fragments. We call it as a discontinuous strand. Only in one direction, that is 5 prime to 3 prime. This is very important. This creates some additional complications. What is that additional complication? See, This is double-stranded DNA. Here we can see hydrogen bonds. Okay. After this point, the hydrogen bonds destroys and it leads to the formation of single strand because it need to undergo DNA replication. And this is the point, we can call it as a replication fork. Now, if you consider this single strand, it is going to act as a template, okay? It is running from 3' to 5'. The polymerization happens in 5' to 3'. So, no problem. It will run continuously. Anti-parallel, right? Out of two strands in the DNA structure, one runs from 5' to 3' direction. The other runs from 3' to 5' direction. So here in this two strands, this is running from 3' to 5' direction and this is running from 5' to 3' direction, anti-parallel to each other. But when compared to here, if the synthesis has to start from here, this strand is already 5', right? This strand is already running 5' to 3'. Again, polymerization has to come from 5' to 3'. It doesn't happen. So here RNA primer addition takes place and starts synthesizing in this direction. Because of this additional complication, it doesn't run in a continuous strand, but instead it runs as a discontinuous strand. And that fragments, we call it as an Okazaki fragments. Okazaki fragments. And these fragments are joined by DNA ligase. Consequently, on one strand, the template with polarity 3' to 5', the replication is continuous. Here, one strand which is acting as a template which runs from 3' to 5', the replication is continuous. Here, we can see continuous strand. While on the other, the template with polarity 5' to 3', 5' to 3', which matches with the direction of the polarization, that is also 5' to 3'. Where is anti-parallel then? It is going to be discontinuous. It is going to be discontinuous. So therefore, this strand, we call it as a discontinuous strand, also called as lagging strand, and continuous strand also called as leading strand. Now, the discontinuously synthesized fragments, what we call it as a Okazaki fragments, are later afterwards joined by an enzyme, and that is DNA ligase. These are all important enzymes. DNA dependent DNA polymerases for polymerization of the new strand. DNA helicases for uncoiling, unwinding. DNA ligase for joining up of the Okazaki fragments. The DNA polymerases on their own cannot initiate the process of replication. It requires origin of replication. Origin of replication. Also, the replication does not initiate randomly at any place in the DNA. It needs a particular site, particular spot, what we recognize as an origin of replication, in short ORI. There is a definite region in Escherichia coli, E. coli DNA where the replication originates, and that is called as origin of replication. Another important point. Because of this reason, Escherichia coli is used as a vector in the case of rDNA technology. which is incorporated with a gene of interest. And the gene of interest has to get doubled if the origin of replication is present, because it need the DNA replication to initiate, it needs an origin, and that origin is nothing but a replication spot, origin of replication. It is because of the requirement of this origin of replication that a piece of DNA, what we call it as a gene of interest, if needed to be propagated, that means replication to be happen, foreign DNA or alien DNA or gene of interest, during recombinant DNA procedure, that is rDNA technology, it requires a vector, and that vector is nothing but E. coli. E. coli. Now, the vectors will provide the origin of replication. That is what we use in rDNA technology, we use this Escherichia coli as a vector. Further, not very details of the replication is understood well. Still, research is going on to understand each and every details of the DNA replication that is happening in both prokaryotic cells and eukaryotic cells. By now, we know that the process that is happening in the prokaryotic cell is simpler when compared to the process that is going to be happening in the eukaryotic cell. Reason is simple, because the eukaryotic cells are much more higher organization when compared to the prokaryotic cells. In eukaryotic cells, the replication of the DNA takes place in the S phase, synthetic phase. We know that S phase and the G phase, gap phase, will be present in each and every cell cycle, and this replication of the DNA happens in the synthetic phase, that is S phase. The replication of DNA and cell division. Replication as well as cell division has to be synchronized, should be very highly coordinate with each other, such that we can eliminate the chromosomal aberration conditions. So here, see, DNA replication has to be coordinated with the division of the cell, such that each cell will get a correct number of chromosomes. That means a failure in the cell division, if the DNA replication happens but the cell fails to divide, then what happens? The dividing both the two daughter DNA will be present in the single cell itself, which leads to a failure in the cell division after DNA replication, results in polyploidy, and that condition is called as a chromosomal anomaly, chromosomal aberration, which consisting of more than two sets of chromosomal number. Now, just now we said that using diagram. So this is the parent DNA. Now this parent DNA is going to separate, show a small opening, that is replication fork, which shows two single strand, and the strand which has a polarity of 3' to 5', where the polarization happens in a 5' to 3' direction. Therefore, it leads to the formation of continuous strand, continuous synthesis. Whereas an other strand which runs with the polarity of 5' to 3', there the strand shows some complications. That is why it is formed in an Okazaki fragments, and we can say this is discontinuous synthesis. And these Okazaki fragments further join together by means of DNA ligase. So this is an picture which represents a complete DNA replication. Just now we said the parent DNA, which is coiled, which is present helical, it is first going to unwind. Uncoiling takes place, and for that uncoiling we need DNA helicase enzyme. And for the stability of the single stranded, see, at one point a very small opening we do find, and that opening, what we call it, is a replication fork. In the replication fork we find the presence of single strands. This is one single strand and this is another single strand. To maintain the stability of this single strand, we need a protein, and that protein is single strand binding proteins. These proteins will bind to the single strand DNA and provides a stability to the single strand. On the either sides, we have the single strand binding proteins. Now, after that, we have two strands here. Here we can see it is running from 5' to 3' direction. And therefore, the synthesis has to take place from here 5' to 3', which is continuous. And here is a DNA polymerase enzyme, which is useful to add the nucleotides in the newly synthesizing strand, taking the parent strand as a template strand. Coming to the other strand, which runs in the direction of 5' to 3'. So here it has to start. And this goes in a discontinuous fashion, which leads to the formation of fragments, and that fragments called Okazaki fragments. And these Okazaki fragments connects to another Okazaki fragment by means of an enzyme, and that is called as DNA ligase. For the polymerization to initiate, it requires a DNA primer. It requires an RNA primer, and that RNA primer is produced by an enzyme, DNA primase. So here is an enzyme, DNA primase, which produces primer of RNA type, RNA primer. And this is a DNA ligases, which attaches the fragments, and that fragments are Okazaki fragments. And here is a continuous strand, which is otherwise called as leading strand. And here is a discontinuous strand, which is otherwise called as lagging strand. So this is all about a complete picture of DNA replication. Right. So with this class, we finished the DNA replication. In the coming up class, let's take up a transcription topic. Until then, keep learning with We Learning. Thank you.